welcome to the Pharmacopause friends. Plasma membrane, as you all know, it is a dynamic fluid structure and it forms the external boundary of the cell. It acts as a selectively permeable membrane and regulates the molecular traffic across that boundary. There are several models out there which can explain the plasma membrane, but the most common model, most accepted model, is the model proposed by the Singer and the Nicholson, the fluid mosaic model, which is proposed in 1972 and which is now the most accepted model. And it is a model of the membrane are viewed as a quasi-fluid structure in which the proteins are inserted into the lipid bilayer. It describes the both mosaic arrangement of the proteins embedded throughout the lipid bilayer as well as the fluid movement of the lipids and the proteins alike. As you can see here in this figure, it is the lipid bilayer with integral proteins and the carbohydrate. As you all know, mainly the lipidic bilayer, proteins and the carbohydrates are there, and this three compose the plasma membrane. And the chemical constituents of the plasma membrane differ from membrane to membrane. For example, the human RBC contains the 43% of lipid and 49% of protein by weight, and whereas the plasma membrane of the liver cells contains 54% of lipid and 46% of protein. So here, and the carbohydrate, the third part is the carbohydrate, which comprise only 5 to 10% of the membrane mass. So first of all, I would like to go for lipids, which is mainly of three types: phospholipids, glycolipids, and the sterols. And the proteins are of integral proteins, peripheral proteins, and lipid-like proteins. While in case of carbohydrates, the protein-like is there. And first of all, lipids. As we all know, the basic structure of the plasma membrane is the lipidic bilayer, and this bilayer is composed of the two leaflets of amphiphilic lipid molecules. Those polar head groups are in contact with intra and the extracellular aqueous phase, whereas the non-polar tail faces each other and constituting the hydrophobic interior of the membrane. The primary physical forces for organizing the lipid bilayer are the hydrophobic interactions. Three classes of the lipid molecules are present in the lipid bilayer. They are namely the phospholipids, glycolipids, and the sterols. So first of all, the phospholipids. Phospholipids are made up of the four components: an alcohol, fatty acid, phosphate, and the alcohol attached to the phosphate. As you can see here, there's the alcohol, phosphate, glycerol, and the fatty acid, which is attached to the alcohol. Now, the fatty acid components are the hydrophobic, while remaining all the components are the hydrophilic in nature. There are two types of phospholipids, namely glycerophospholipids and the sphingophospholipid. As you can see here in the slide, it is the glycerophospholipid. Phospholipids are derived from the glycerol, are terms of glycerophospholipids, and the glycerophospholipids contain glycerol, fatty acid, phosphate, and the alcohol. If the alcohol is choline, it is termed as the phosphatidylcholine. If it is ethanolamine, it is termed as the phosphatidylethanolamides, and so on for serine and inositol. And for sake of knowledge, phosphatidylcholine is also known as lecithin, while phosphatidylethanolamin is known as the spirulina. While second type of the phospholipid are the sphingospholipids. Here the simple change is that alcohol is the sphingosine instead of the glycerol. Sphingolipids contain the amino alcohol named, it is the amino alcohol sphingosine, and the other structure remains the same: fatty acid, phosphate, and the alcohol attached to the phosphate. In sphingophospholipid, the amino group of the sphingosine backbone is linked to the fatty acid by amide bond, and the sphingomyelin is the most abundant sphingophospholipid. The plasma membrane of the animal cell mainly contains four phospholipids, and they are namely phosphatidylcholine, phosphatidylethanolamine, phosphatidylserine, and the sphingomyelin. At on the neutral pH, pH 7, the phosphatidylcholine and the phosphatidylethanolamine doesn't have any net charge, while phosphatidylserine is having the net negative charge. And next, coming to the glycolipids. Glycolipids contain the carbohydrate which is covalently attached to the lipid. This can derive from the either glycerol or sphingosine. The simplest glycolipid is the saccharose, which contains the single sugar residue. Sugar can be glucose or galactose. Glycosides are the more complex ones, which consisting of the branch chain of the sugar, may be 7 or more than that. And the glycolipids are mainly found on the surface, and the surface, which is the outer leaflet of the plasma membrane, with their carbohydrate portions exposed on the cell surface. And the sterols mainly contains the cholesterol, mainly the cholesterol is there, which act as the fluidity buffer. And apart from that, lipid bilayer having many other functions, that it having the lipid raft, and how the micelle, reverse micelle, and the bilayer formed, asymmetry of the lipidic bilayer, and the motion of the lipidic molecules, and the fluidity of the lipid bilayer. Lipid raft mainly composed of the phospholipids and the cholesterol, while micelle, reverse micelle, and the bilayer, it is based on the critical packing parameter of the lipid taking part in the formation of the lipidic bilayer. While in case of the asymmetric lipidic bilayer, it is due to the distribution of the phospholipids through outer part and the inner part of, means the outer part means the external part, and the inner part is the cytosolic side of the membrane. While in case of the motion of the lipids, it can be the rotational motion or the lateral motions. While in case of fluidity of the lipid bilayer, it mainly depends on the lipidic composition and the temperature. And the next part is the proteins, which are the proteins present in the plasma membrane. Proteins mainly the peripheral, integral, and the lipid-linked protein. As you can see here, the cytosolic side and the exoplasmic side. Here's the lipid anchor protein, which is having the lipidic linker in between the plasma membrane and the protein part. While integral protein, it is embedded in lipidic bilayer, and the peripheral protein, either they are on the lipidic membrane or maybe attached to the protein itself. So in case of the first of all membrane proteins are responsible for the most of the dynamic process carried out by the membranes. The most membrane proteins are classified as peripheral or integral, and lipid linked is the small part. Many of the proteins associated with membrane can be released from the membrane by relatively gentle extraction procedure, and these are termed as the peripheral protein. While some proteins which are bound to the very tightly to the plasma membrane, they are termed as the integral proteins. Generally, the transmembrane proteins, they are composed of the nonpolar amino acids. They are the integral proteins. While in case of the peripheral proteins, electrostatic and the hydrogen bonding interaction are the main interaction. While they do not act by means of the hydrophobic interactions. For example, pectin and ankyrin, these two are the peripheral proteins which are responsible for the shape and the flexibility of RBC. While all the transmembrane proteins are amphiphilic in nature, and they are the example of the integral proteins. Glycoprotein is the major single pass transmembrane protein present in RBC, which is about of 131 amino acid, and it is the first transmembrane protein for which the complete amino acid sequence was determined. And despite of their abundance, it is still unknown that what is the function. So here peripheral protein, they are generally water soluble, and electrostatic and the hydrogen bonding are the main interactions. Pectin and ankyrin, as I told you, they are present in RBC. Integral proteins, they are made up of nonpolar amino acids, and they can be of single pass or multi-pass. Single pass, as I told you, glycoprotein is the single pass, while all transporters are the multi-pass proteins. Next is the lipid-linked proteins. Lipid-linked proteins, they are covalently bound to the lipid molecules, so they are called lipid-linked proteins. They are form covalent attachment with 3 classes of lipid. On the basis of that, they may be the lipid class, maybe the isoprenyl group, or maybe the fatty acid. Here, in case of the isoprenyl group, the attachment site is the cysteine residue of the C terminus of the amino acid. And in case of the fatty acids, they may be the myristic acid or palmitic acid. And in case of myristic acid, the glycine residue at on the N terminus, as you can see here, the fatty acid myristic acid, the glycine at the C terminus with amide linkage, while in case of palmitic acid, it binds to cysteine residue at on N or C terminus with a thiol linkage. And this all isoprenoid and the fatty acid, this two type of fatty acid, myristic and palmitic fatty and the isoprenoid, are there on endoplasmic phase. While in case of the glycosyl phosphatidyl inositol molecule, it is bind to the amino acid at the C terminal with the lipid linkage, and it is on the exoplasmic phase. So here the last part is the carbohydrate present in the plasma membrane. As I told you, carbohydrates are very less, they are mainly present on the surface, and these carbohydrates are oligosaccharides, occur both as a glycoproteins and the glycolipids. And the mainly present carbohydrate is the glycocalyx or the cell coat, which protect the cell from the mechanical and the chemical damage, and it is also useful in the cell adhesion. So this is all about the plasma membrane. Thank you.